Okay, I think we can start. I've seen that people taking coffee have no intention of coming here. So, good morning, everybody. They're coming? They're coming with the coffee. It's okay. You could have come with the coffee. So, good morning, everybody. Thank you for coming to this meet and greet, live or connected. It's a pleasure for me to introduce to you Dr. Miruna Popa. She's a cardiologist, a fellow researcher, coming from the German Heart Center in Munich in the group of Isabel Deisenhofer, which some of you might know as a partner of certain clinical studies, including BTF, ongoing at the Institute. And she's coming here for 1 year of research as a visiting fellow, and she will be under the coordination of Pierre Jaïs for what concerns her project. And you will see that it's a very interesting translational project between the phenotype of atrial fibrillation and the effect of different types of ablation for what concerns some very peculiar and interesting inflammatory markers. So that being said, I'm leaving the stage to Miruna. Please.
Thank you, Guido, for the kind introduction. Hello, my name is Miruna Popa, and it is with great pleasure that I'm able to meet you all today. Thank you for coming, and I'm going to show you some slides on my research background and my clinical experience. So I graduated from Heidelberg Medical School in Germany. This is where I obtained my medical degree in the division of cardiovascular physiology. It is in this lab that I acquired experience mainly concerning blood cell isolation and flow cytometry, platelet function testing, and the analysis of platelet monocytes interaction under flow conditions. Afterwards, I started working as a medical resident in the Department of Electrophysiology at the German Heart Center in Munich. And this is where I gained clinical experience in the management of atrial fibrillation. Actually, starting 2019, I've been a member of the EP lab there, and I was able to perform RF ablations, mainly of AF and AT. I was also part of management of clinical trials, so I gained experience in that field as well. Starting my experience with basic science, so I'm going to show you a slide on my medical thesis. It analyzed basically the mechanism of monocyte recruitment to endothelial cells in the context of atherosclerosis. It is known that monocyte recruitment and infiltration into the arterial wall is a driving force of atherosclerosis. However, it was widely unknown how monocytes can overcome arterial shear stress in order to firmly adhere to endothelial cells. So our hypothesis was that platelets can mediate those monocyte endothelial cell interactions, and that platelets binding to ultra-large von Willebrand factor strings, as we see here in this image. It is known that activated endothelial cells can secrete this von Willebrand factor, which can form ultra-large strings to which platelets adhere as bead-like structures. This process does not normally occur under physiological conditions because the specific von Willebrand factor protease, ADAMTS13, cleaves those von Willebrand factor strings, rendering them inactive. However, under certain pathophysiological conditions and chronic inflammatory conditions such as atherosclerosis, it is known that ADAMTS13 levels can be decreased. So in order to test this hypothesis, I established flow model in vitro using human endothelial cells, which were plated in a parallel plate flow chamber. And I did perfusion experiments using fluorescently labeled platelets and monocytes. And as we can see here in those images, we were able to show that monocytes stained in blue preferentially interact with platelets, forming these bead-like structures on the surface of endothelial cells, and that platelets seem to play a major role in monocyte recruitment to activated endothelial cells. We were also able to show that the CD40 ligand interaction plays a major role in this mechanism. We were also able to show that ADAMTS13 expression levels were decreased when incubating endothelial cells with inflammatory cytokines. And we found in a small clinical study, we found low ADAMTS13 plasma levels in coronary artery disease patients. And so basically high levels of von Willebrand factor, low ADAMTS13 plasma levels, and platelet monocyte interactions can represent an important link between thrombosis and inflammation. I will move on to my clinical research. So after starting work at the German Heart Center, I still remained interested in inflammatory conditions, and I studied the significance of early recurrences of atrial arrhythmia following catheter ablation, as it is known that inflammatory response post-ablation is a driving force of early recurrences. And we were able to find that early recurrences have certain or a significant predictive value in predicting late recurrences. So here patients without early recurrences, here it's the blue curve, have a significantly better outcome after one ablation and also after multiple ablations. Furthermore, another recent study which I performed has been recently accepted for publication, EP-Europace. It studied the safety profile and long-term efficacy of very high power short duration using 60 to 70 watt catheter ablation for atrial fibrillation. I'm honored and glad that I'm going to show the results of this paper in the ongoing or coming Journal Club on the 8th of November, and I'm not going to spoil the results now. We'll be able to discuss them in detail on November 8th. So coming to an end, what is my current research project at Lyric? It's basically, since I'm aiming to combine my basic research experience in cardiovascular inflammation with my clinical experience in atrial fibrillation, and I'm interested in studying the role of inflammation in the pathophysiology of atrial fibrillation. One project aims at analyzing the inflammatory response following pulsed field ablation compared to conventional RF ablation. In order to test this, we will be having a look at biomarker analysis in patient blood samples before and after ablation. We will be looking at inflammatory cytokines and platelet monocyte complexes using flow cytometry, and we will also analyze platelet activation markers. So we want to know whether we also see in the clinical setting that PFA leads to a decreased inflammatory reaction. And we want to test in a further sub-study whether this translates into reduced incidence of early recurrences. Also, it is planned that we will be having a look at histological analysis of PFA lesions in an animal model. And in a further project, we would like to define the mechanisms linking inflammation to AF substrate formation. It is planned to characterize the inflammatory response in a sheep model of AF progression using already available samples. And this will be done in a first step by doing a histological analysis of inflammatory infiltrates and analyzing plasma markers of inflammation and platelet activation. So I would like to thank you very much for attending, and I'm glad to answer any questions.
Thank you, Miruna, for this presentation. My opinion on small opinion on this study that you are including. I think your experience brings an important added value both to the pathophysiological work and the clinical work that we are doing here. Circulating biomarkers are incredibly valuable to define better both the approach to have certain arrhythmias such as atrial fibrillation and understand better the mechanism that underlie this pathology, which frankly we don't yet understand properly. If there are some questions from the audience or online, please go ahead.
Thank you for the presentation. Could you elaborate a bit on your findings between early and late AF recurrences, about why this isn't obvious?
Yes, so we know current guidelines say that early recurrences, that there's a blanking period of 3 months should be considered following ablation, during which early recurrences are not considered to have to be of prognostic significance. But there is increasing data on this, not only my paper, but also other papers in paroxysmal and also persistent atrial fibrillation, which have shown that early recurrences actually do have a prognostic significance. And there's also a discussion ongoing whether a blanking period of 3 months is actually validated. There are results showing that maybe 4 or even 4 to 6 weeks is actually sufficient for this blanking period, because every recurrence occurring after that period is almost equal to a late recurrence. So there is certain also discussion on the mechanism of early recurrences. So one is certainly in the inflammatory response. Another possible mechanism can certainly also be pulmonary vein reconnection. In the context of persistent atrial fibrillation, I'm not sure if that is the driving mechanism. I also think that substrate, so pre-existing substrate, can also play a role, whether it's acute inflammation, which is done after ablation, but also chronic inflammation leading to that substrate. I hope this answers your question.
Could I summarize? If it recurs, it's not because the doctor just screwed up the ablation, just because they have a lot of possible surface.
Yes, exactly. And one hope would be if we were able to show that new ablation technologies such as PFA, where we know that it induces more homogeneous lesions and the architecture of the tissue is preserved, that this would also lead to a lower rate of inflammatory response at the atrial tissue level, but also on systemic level. And this may translate into less early recurrences, which would definitely have an impact of the quality of life of patients, because for them, as having a recurrence 2 days or 1 week after ablation, it's not such a nice experience. And it could also have an impact of hospitalizations. Thank you.
Yeah, thanks again. So I sat in a symposium a couple of weeks back where each of the clinicians kind of had their own frequencies of how they apply the PFA. It doesn't seem too standardized how they apply the PFA. So is the inflammation consistent always based on what the actual clinician has done, in a sense where the acute inflammation, is it dependent on the magnitude of the PFA that they're applying, the amount of PFA they're applying, or just the amount of PFA they're applying, and there's automatically acute inflammation afterwards that these patients get. where I'm getting this, what's kind of confusing probably is just the fact that I think there's so many dials will make PFA. They can control so many different things. So
Yes.
is the information consistent every time?
Yes, this is a very good question, and the answer is we don't know yet, and that's why I think there's a need for more research in that direction. And we know from histological studies that there is less inflammation, but we don't really have much. It hasn't really been characterized so well so far as I know.
Just follow this. So do you notice the difference between how PFA is applied here and also here versus in Germany and wherever you have seen different centers applying PFA? Is it pretty standardized or is it pretty?
Well, we know there's different catheters. Now we are in Germany. We started using the Farapul system, and this spring, and we're also going to participate in the BTF trial using this Farapul system. And as far as I know, this is also used here, so this will be equivalent. But we started using, actually it's quite recent, 6 weeks ago, if I'm not mistaken, a new technology also using PFA from Galaxy Medical, which uses point-by-point lesions. And there we saw that it makes a huge difference. Because when using Farapulse, one gives many impulses on the same spot. And when using point-by-point, you have to adapt. It makes a difference, certainly. And the technicians also showed us some very interesting data that there's also a myocardial stunning after that. And yes, so I completely agree. PFA is still quite heterogeneous, and we will have to define which PFA protocol leads to which cellular inflammation. That needs more research, certainly.
Any reason on the topic of different modalities of inflammation? The basic science is not there, of course, as you can imagine. And this is why it's so important to study it further. You can imagine RF ablation. So the cell death induced by RF ablation is considered necrotic. So necrotic death is an irregular opening of plasma membranes, release of nucleic acid, intracellular organelles, DNA. This creates a cascade of acute inflammation, which is very well known. It's typical. It's very similar to wound healing. But the assumption on the physical principle of PFA is that this works on irreversible electroporation. Irreversible electroporation works by creating nanopores which are thermodynamically stable. So the extrusion of intracellular material might be less evident, and a big assumption in certain levels of electroporation, and it's been observed as well in certain tissues like cancerous tissues, that most of the cell death is mediated by apoptosis, at least a little bit far from the electrodes. Very close to the electrodes, we have increase of temperature, we have very high electric field, so we can assume that to some extent it resembles necrosis, at least for the extrusion of intracellular material. But a little bit far from the electrodes, probably the cell death is mostly apoptotic. And this allows the immune system to manipulate the tissue and to clear the tissue more organically, you can say. And this is thought to preserve the thickness of the tissue. This is not true all the time. Like if you see the first experiments on electroporation performed by the group of Karl Sneven and Fred Wittkampf, where they used a lot of energy, like 300 joule of energy on the ventricle tissue, you can see the typical shrinking that is also visible in RF ablation. So it's really a matter of how you use it. Whereas after 30 years of experience, we know that 60 degrees of increase in the tissue triggers thermal cell death. If you go lower, you need a certain amount of time for this temperature to be kept in order to trigger cell death. For electroporation, cell death may take hours, and it might be composite. The Farapulse catheter has one strategy. It uses a large amount of energy to ablate in one shot, this one-shot technique. The Sphere 9 catheter, the Afera system, uses a different strategy from the basket catheter. So it relies on flexibility. And I don't have a lot of experience with the Galaxy Medical. Point-by-point ablation is very similar to RF ablation as a concept. So it needs another kind of standardization. We're not talking about thermal fields here. We're talking about electric fields, so orientation, density, duration. This all changes how your tissue sees the damage and receives the damage. There's also the huge problem that we don't know if, for example, if there are monocytes infiltration in the fat, which mediate these inflammatory processes that might promote AF substrate, PFA might not be effective for this because fat is a perfect electrical insulator. There's a plethora of data done on the thigh of the pig that shows that if you choose a fatty area, then electroporation is impaired, which is not as much for thermal energy. You can increase the temperature and get some effect. So it's really not standardized, and the research is needed in every level.
I found this interesting too, that they still don't know a lot about the underlying substrates and the material differences, where I think your sheet level, fat everywhere, dry differences and stuff. So you want to
a little bit venture into the evolutionary biology of the thing. I still don't understand why sheep have a fatty right atrium, but a lean left atrium. Why? What's the advantage? But it creates a perfect control of the same animal.